Bioinfocongress IV Assistant Professor Dr. Defne Bayık Consideration of Biological Sex for Immunotherapy of Glioblastoma. Assistant Professor Dr. Defne Bayık is a cancer immunologist focused on uncovering host regulatory pathways as targets for brain tumor immunotherapy. She completed her undergraduate degree in the Bilkent University Department of Molecular Biology and Genetics and received her PhD in 2016 through the National Institute of Health, Bilkent University Graduate Partnership Program. Her dissertation work, under the mentorship of Drs. İhsan Gürsel and Denise M. Klinman, identified differentiation pathways of immature myeloid cells to inflammatory and immunosuppressive macrophages. In 2017, Dr. Bayık joined the laboratory of Dr. Justin D. Latia at the Cleveland Clinic as a postdoctoral fellow, where she gained experience in neuro-oncology and translational cancer immunotherapy research. She demonstrated that sex differences in the anti-tumor immune response inform targeting of immunosuppressive myeloid cells in high-grade brain tumors. Her postdoc work also defined epigenetically regulated pathways that inform the functional heterogeneity of myeloid cells. She joined the University of Miami Silvestre Comprehensive Cancer Center as an assistant professor in December 2022.
Dear Dr. Defne Bayık and participants, welcome all of you to our Bioinfocongress IV event. Dr. Defne Bayık will give us a talk today on consideration of biological sex for immunotherapy of glioblastoma. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dr. Defne Bayık, welcome back.
Betül, thank you very much for that very kind introduction, and I thank you very much for your invitation to give this talk. I'm very excited to be here with you guys virtually and looking forward to connecting afterwards as well. So as Betül alluded to, I am a very new assistant professor who started I started my lab just 3 months ago, and we are interested in understanding how biological sex could be an important factor determining immune response in brain tumors and how we can take advantage of that for immunotherapy of glioblastoma. So just to briefly outline my presentation, I would like to very briefly introduce the role of the immune system in glioblastoma in brain tumors. I would like to briefly recap some of my postdoctoral work, which focuses on sex differences in myeloid-derived suppressor cells and how epigenetics is important for their regulation. And I would like to talk about 3 ongoing projects in my laboratory that focuses on myeloid cell regulation and aims to identify new therapeutic targets for brain tumors. So what is glioblastoma? Glioblastoma, or in short GBM, is grade 4 glioma. So this is the most common primary malignant brain tumor. This is a very aggressive brain tumor, and the standard of care has not changed over a decade. When we look into what constitutes standard of care, it is surgical resection, followed by radiotherapy and chemotherapy, which is temozolomide. And these treatment modalities lead to a median survival of somewhere around 14 to 16 months. More recently, there are other treatment strategies that are getting incorporated into glioblastoma treatment, and one such strategy is tumor treating fields that manage to extend the survival duration up to 20 months. So this is still not a great outcome for these patients. There is a growing appreciation for the use of immune therapies in many solid and hematological malignancies, but these strategies did not lead to a good therapeutic response in patients with glioblastoma. As many of you are aware, anti-PD-1 and anti-CTLA-4 and the scientists who discovered these molecules got Nobel Prize a couple of years back, are now becoming standard of care. But these 2 reagents did not lead to any extension of survival in patients with glioblastoma. and the response rate is somewhere around 2%. So there is really great need to understand what immune mechanisms are involved in treatment of glioblastoma and application of immune therapies. And there are several considerations for that. So this is a tumor of brain, so there is blood-brain barrier that prevents effective accumulation of therapeutics in brain tumors. There are stem cells that lead to therapeutic resistance, and after surgical removal, the presence of these cells lead to recurrence. But there is also the immune system. So if we understand what kind of immunological changes are happening in the tumor microenvironment, we can target these for effective treatment strategies. And if we look at glioblastoma, there are many immunological changes. So these are data from patients, and what we observed was that patients with glioblastoma had very low levels of T cells, almost equivalent of patients with HIV. So these T cells usually end up in bone marrow, sequestered in bone marrow, and this is induced by the tumor itself, as well as the treatment, the chemotherapy we use in patients. In the tumor microenvironment, what we observe is accumulation of immune suppressive type of cells, or exhausted T cells that cannot fight with tumors effectively. Glioblastoma is not unique, but it's different in the sense that there is very heavy myeloid cell presence. So if we again look into the tumor microenvironment, there are resident microglia cells, tumor-associated macrophages, and my population of interest, myeloid-derived suppressor cells. Collectively, these three myeloid cell populations can constitute up to 30 to 50% of tumor bulk. So there are almost as many immune suppressive myeloid cells as tumor cells in this microenvironment. And when I joined my postdoctoral fellowship, we had studied the role of myeloid-derived suppressor cells and patient outcomes. And what we observed was that in patients, if there's a reduction in the frequency of myeloid-derived suppressor cells after initial treatment, these patients have tendency to do better. If we observe an increase in the frequency of circulating myeloid-derived suppressor cells over time, these patients usually have a poorer outcome. So these studies really defined myeloid-derived suppressor cells as biomarkers of disease and biomarkers of brain tumor in particular. But at the time, we did not really study the individual role of myeloid-derived suppressor cell subsets. So what do I mean by that? So when we look at the MDSCs, there are broadly categorized into two subsets as monocytic MDSCs and granulocytic MDSCs. Both these cells are generated in the bone marrow. They are immature, but they have different properties. We can use phenotypic markers to define these cell populations. There are different markers we use for human and mice, and this helps us to discriminate monocytic subsets from granulocytic subsets, or polymorphonuclear is another name for this subset. We know that both subsets are capable of suppressing T cell response, and that is the functional role of MDSCs, the suppressed T cells. But recent studies suggested that they can undertake additional roles in the tumor microenvironment. Specifically, a study in the context of breast cancer showed that monocytic MDSCs can promote stem cell maintenance, cancer stem cell maintenance, whereas granulocytic MDSCs can promote differentiation, metastasis to distant sites. However, the role of these cells were not defined in the context of brain tumors. So for the purpose of this talk, it's important to know that glioblastoma is a disease in which we observe sex differences. So if we look at the incident rates across age groups, what we observe is that 60% of all glioblastoma patients, or all glioma patients, which are the brain tumors, are males. So this is a male prevalent disease. If we look at the outcome, we observe that male patients have a poor outcome compared to female patients. So what is the basis of that? Looking epidemiologically, this is not unique to glioblastoma. If we look at the various diseases from autoimmunity to cancer, we observe a strong sex bias. Females overall are prone to autoimmune diseases and inflammatory conditions, whereas very non-reproductive cancers such as liver, lung, are more common in males compared to females. And this is something we can also see in immunocompetent models as well, or model in immunocompetent models. So in lab, we frequently use syngeneic mouse glioblastoma cell lines that we implant orthotopically into brains of mice. If we use immunocompetent mice, what we observe is that, and I do realize this data points don't look well, is that male mice, which is I try to highlight with my pointer, succumb to disease earlier than female mice. And this is consistent across different models of glioblastoma, which have different driver mutations. If we repeat the same experiment with NSG mice which lack intact immune response, we observe that there is no difference between the median survival of males versus females, suggesting that, that there is a role for the immune system. So that's what I tried to identify as a postdoctoral fellow. And long story short, these results got published several years back, showed that there's increased accumulation of monocytic subset of myeloid-derived suppressor cells in the tumor microenvironment. And these cells were more abundant in males compared to females. And we observed this both in animal models as well as patients. We observed a role for granulocytic MDSCs in females. Specifically, there was an increased expansion of these cells peripherally in females. And in patients, we observed that high levels of granulocytic MDSC or score for a granulocytic MDSC correlate with a worse outcome. So we wanted to target these differences with the hypothesis that if we target the sex differences, we can get differences in immune therapy response. So we performed gene expression analysis and then combined this with a drug prediction analysis to define targetable pathways. And what we ended up observing kind of validated our hypothesis. So when we targeted monocytic MDSCs non-specifically with a chemotherapy named fludarabine, we observed that male mice with glioblastoma tumors had a better survival, whereas this treatment had virtually no effect on female mice with tumors. In contrast, if we block IL-1 beta, which was mainly secreted by granulocytic MDSCs, this time, there was no outcome differences in males, but in females, there was a significant extension of survival, suggesting that neutralization of IL-1 beta could be a good therapeutic approach for females and not males. But at the time, we really didn't address what drives this differences in MDSC subsets. So that's what I tried to do later in my postdoctoral career by using adaptive transfer approaches. So in this setting, we implanted mice with tumors, and then we adaptively transferred MDSCs to these tumor-bearing mice to measure the survival outcomes. As we were doing that, we used matched sex donor and recipients, as well as donor and recipients from opposite sexes. So the aim of this this design was that by focusing on the donor sex, we tried to identify cell intrinsic regulatory mechanisms, whereas by looking into recipient sex, we wanted to understand host factors or environmental factors that are unique for males versus females. So in this series of Kaplan-Meier curves that I'm going to walk you through, it actually turned out that there was a role for both cell intrinsic and environmental regulation. So the first pair we are looking into here is male donors that go into male recipients. And in this setting, what we observed was that transfer of monocytic MDSCs significantly accelerated tumorigenesis. That is blue line compared to the black line. We didn't observe the same tumor driving effect when we transfer granulocytic MDSCs. That is orange line compared to black line. When we use female donors and put them into male recipients, we observed a similar effect in which monocytic MDSCs drove the disease and accelerated the tumor progression, but the magnitude of effect was much limited. In contrast, when we put these cells into female recipient mice, there was virtually no effect in the survival outcome. So collectively, these experiments suggested that monocytic MDSCs were the tumor driving factors, and it didn't matter whether they originate from a male or a female mice, but they were only able to exert this activity in a male microenvironment. So we wanted to understand what makes a monocytic MDSC a tumor driver compared to granulocytic MDSC. And to do so, these cells are generated in the bone marrow. So we sorted them out and collaborated with a researcher from Case Western University, Peter Scacciari, to perform ataxia. So this is a method in which we can detect open chromatin regions by using linker sequences. So when we did that and both characterized the epigenetic architecture of these cells, we observed that the main driver was cell identity. So what I mean is that in this principal component analysis, you can observe that 70% of the variation in the epigenetic structure was introduced by cellular identity. Monastic MDSCs were clustered separately from granulocytic MDSCs. There was still a role for sex that accounted for 27% of the variation. So since the cell identity was the main driver of the epigenetic profile, we wanted to understand what genes were located in these open chromatin regions in the monastic MDSCs and which pathways they were linked to. When we did this analysis, our top pathway was cell adhesion. So cell adhesion-related genes were more available in monastic MDSCs compared to granulocytic MDSCs. To further validate these observations, we actually went ahead and did a surface flow staining for cell adhesion molecules, which are integrins. And we observed that integrin beta-1 and integrin beta-7 were highly expressed in monastic MDSC subset, but not in granulocytic MDSC subset. We wanted to understand whether this expression had a role. So we did the same adaptive transfer experiments I shared with you earlier. But this time, we blocked the surface integrin beta-1 or integrin beta-7 before transferring these monastic MDSC into recipient mice. So in this setting, the cells get pre-treated and not the mice. And what we ended up observing is that when we block integrin beta-1 on the surface of monastic MDSCs, they lose their tumor driving effect. So this is solid line compared to dashed blue line. In contrast, when we block beta-7 on the surface, we don't abrogate their tumorigenic function. These results suggested that integrin beta-1 could play a role in the trafficking of monastic MDSCs, or at least their function as tumor-promoting cells. So, as I briefly mentioned earlier, monastic MDSCs are immature cells that are capable of differentiating into macrophages or dendritic cells when they encounter right cytokines. And since macrophages are very prevalent in the tumor microenvironment, and tumor-associated macrophages are actually main drivers of immune suppression, we wanted to understand whether by blocking integrin beta-1, we were abrogating their differentiation into macrophages. So again, these are the adaptive transfer experiments I shared with you earlier. We color control mice and we transfer isotype treated monastic MDSCs or monocystic MDSCs that were pre-treated with an anti-integrin beta-1 blocking antibody. When we look at the frequency of different myeloid cell populations, immature cell subsets on top and more differentiation subsets in the bottom, what we observed is that when we transfer isotype treated monastic MDSCs, there's an increased prevalence of macrophages. Whereas when we block integrin beta-1, this effect is diminished, and in fact, we actually observe more dendritic cell subsets. So this really suggested that we are abrogating monastic MDSC macrophage communication by blocking integrin beta-1. These are all preclinical studies, and obviously we always have in mind to translate these observations, basic observations, into the clinic. So we wanted to therefore understand whether integrin beta-1 expression and these differences we observe in mouse are relevant to patients. To do so, we actually turned into publicly available single-cell data sets and created signatures for monastic MDSCs or granulocytic MDSCs. Then we compared the integrin beta-1, beta-2 as a control, and beta-7 expression levels between these two subsets. So the patient data kind of validated what we were observing in preclinical models. Both integrin beta-1 and beta-7 expression levels were higher in the monastic subset compared to the granulocytic subset. We also confirmed this by performing flow cytometer staining. We got patient specimens, blood and tumor specimens from patients with glioblastoma and stained for integrin beta 1 and beta 7 on the surface of the cells. We observed that there were very high levels of integrin beta 1 expressed on the monastic MDSCs compared to granulocytic MDSCs. So, to our surprise, integrin beta 7 was not highly expressed by either cell population, and it was not different, which was kind of controversial compared to the RNA expression levels, but we did validate this with 2 separate antibodies. So this really is a take-home message that RNA and protein don't always correlate. But collectively, combined with the mouse data, these results suggested that integrin beta 1 could be a good candidate to target monastic MDSC activity in patients with brain tumors. The only problem is that integrins are very cardinal. They have a very central role in human physiology. So though there are inhibitors and though they are in clinical trials, they are not the best drug candidates. Therefore, we went back to our cell adhesion signature and tried to understand what other genes were related to integrin beta 1. And one such gene was dipeptidopeptidase 4, or DPP4 in short. You can appreciate that if you look at the peak graphs here. These peaks are more prominent in monastic MDSCs compared to granulocytic MDSCs, suggesting that this gene is getting actively transcribed. And in fact, when we look at the RNA level, we see that monastic MDSCs have significantly higher levels of DPP4 expression compared to granulocytic MDSCs. And we also wanted to again validate this in patients and looked for their expression levels in patient monastic MDSCs versus granulocytic MDSCs using the single-cell sequencing data sets. And again, DPP4 was highly expressed by monastic MDSCs, but not granulocytic MDSCs, which is also shown here. So this top purple part is the monastic MDSCs, whereas this bottom gray part is the granulocytic MDSCs. So is DPP4 a relevant target? And it appears so. If we look at the expression levels across tumor grades using the Cancer Genome Atlas, we observed that patients with grade 4 tumors, which are glioblastoma, had significantly higher levels of DPP4 expression compared to patients with lower grade tumors. Furthermore, only in the glioblastoma subset, if we look whether DPP4 is predictive of survival, we observed that high levels of DPP4 correlated with worse outcomes, suggesting that DPP4 might have a tumor-promoting effect. So what is DPP4 or CD26? So this is a multifunctional protein that is present on the plasma membrane. There's also a soluble version of DPP4 that is secreted from these cells. So on the surface of the cells, DPP4 forms interactions with different kind of proteins, including integrins, and activates ERK phosphorylation pathway. Soluble DPP4 is important for regulation of glucose metabolism. DPP4 is an exopeptidase which has ability to cleave cytokine, chemokines, and incretin hormones. And this cleavage through incretin hormones is important for glucose-insulin balance. And in fact, there are many clinically approved inhibitors against DPP4 that are used in patients with type 2 diabetes. So there are very commonly used drugs that can target DPP4, and they relatively have good safety profile. So we were very excited about that because the standard of care of glioblastoma, as I mentioned earlier, has not changed over a decade. So the main treatment approach is to do clinical trials. And if we have drugs that are readily available in clinic, we can repurpose them for glioblastoma use. That's why we wanted to understand whether targeting DPP4 could lead to therapeutic opportunity for glioblastoma, and we turned into our syngeneic models. So again, if we implant immunocompetent mice with a syngeneic model, GL261 in this case, and treat them with a DPP4 inhibitor sitagliptin, we observe that these mice that receive drug have a much better outcome compared to those that get the vehicle. If we do the same experiment in immunocompromised animals, we fail to extend survival by any means, suggesting that the effect is likely through regulating the immune response. And in fact, when we analyzed the tumors from this vehicle versus sitagliptin-treated mice with flow cytometer, we observed that there was a reduction in tumor-associated macrophages. So what else? Like, is this related to myeloid cells? It doesn't appear to be so. So these are more recent studies that we are currently pursuing. What we observe in vitro, at least, is that if we treat CD8 positive T cells with sitagliptin, there's an increased proliferation. So there's a potential that we are activating T cells as well. But when we looked into tumor microenvironment, that's not what we observed, because there was still slight reduction in the frequency of CD8 and CD4 T cells. It wasn't statistically significant. But when we also look at the activation status by measuring the levels of CD69, which is a marker of T cell activation, and PD1, which is the checkpoint, a marker of T cell exhaustion, sitagliptin treatment did not change the expression levels. So then the effect is, at least the anti-tumor effect we see is mainly through myeloid cell modulation. And it appears to be the case. So to better understand the outcomes, we did a migration assay to see whether we are actually preventing cells from reaching to the tumor microenvironment. And in this in vitro migration assays, we observed that sitagliptin treatment significantly reduced the migration of monocytic MDSCs towards tumor cells. We also observed that there was a reduction in the phosphoric levels in monocytic MDSCs treated with sitagliptin, which kind of shows us that the drug is effective. So our current studies are trying to understand what DPP4 does in the tumor microenvironment. I mentioned to you earlier that it's an important molecule that regulates insulin glucose metabolism. So in this tumor-bearing mice, we looked at unfasted insulin and glucose levels, and we didn't observe any change with sitagliptin treatment, which is to be expected because this drug only works in patients with aberrant insulin glucose metabolism, and it doesn't necessarily change the baselines in healthy individuals or individuals with no metabolic diseases. We also did with a targeted cytokine analysis from plasma of these mice, and we observed that there was a reduction in the CXCL5 levels. So CXCL5 is a chemokine that involves recruitment of immune cells, granulocytic cells, and macrophages. And in patients, what we observed was that high levels of CXCL1 correlated with worse outcome compared to patients with low levels of CXCL5. So in conclusion, these published and unpublished results suggested that there was an epigenetic regulation that affected the behavior of myeloid cells. Specifically, we observed that the monocytic MDSCs had increased cell adhesion pathway availability, and this led to higher levels of surface integrin beta-1 and DPP4 expression. When we block integrin beta-1 or DPP4, we were able to reduce the frequency of tumor-associated macrophages in preclinical models. So our current research focuses on understanding the role of DPP4 in myeloid cell regulation, and also trying to understand the role of CXCL5. So CXCL5 is a ligand for the receptor CXCR2, and we want to understand whether, if we block this axis, whether we can further alter the immune microenvironment and get favorable outcomes. So what else we are working on? So we are, I mentioned to you that there is a role for intrinsic regulation and environmental factors. So these results that I shared with you were intrinsically, epigenetically making monocytic MDSCs more proteinogenic. But what are the environmental factors that contribute to differential MDSC subset activity? So there is a growing appreciation for the involvement of the neural system in regulation of tumor genesis. This is especially a very hot field in the context of brain tumors, but there's new research suggesting that this is also applicable to peripheral tumors. So several groups, Michel Monger's group and Frank Winkler's group, has shown that tumor cells can integrate into neuronal networks and can respond to electrochemical cues. Furthermore, factors secreted by neurons or other neural cells, such as astrocytes, can alter the tumor cell behavior. But what is not known in this field of cancer neuroscience is that what role the immune system plays. And there is a reason why we are interested in, because several studies in inflammation and autoimmunity have suggested that neurotransmitters can regulate the behavior of immune cells. Specifically, these immune cells were shown to express receptors for various neurotransmitters. And if we alter the neurotransmitters, we can control the inflammation. And when we had done gene expression profiling of monostic MDSCs and granulocytic MDSCs, which led to the identification of drug candidates that I shared with you earlier, one of the top differentially expressed genes between these two subsets was GABA receptor B. So GABA receptor B is a G protein coupled receptor, so it has a signaling function. These cell subsets were not expressing GABA receptor A, which is the ion channel. And in fact, when we looked at the drug prediction algorithm and tried to understand which drugs can readily affect the behavior of granulocytic MDSCs, one of our top drug classes was GABA modulators. So this was very interesting to us and kind of brought up the idea whether we can modulate GABA levels to control the activity of granulocytic MDSCs. One thing before I move into my next slide is that this drug prediction algorithm does not show directionality, meaning that these drugs do not necessarily block the function of granulocytic MDSCs. So that's what we tried to do, and we were very optimistic and hoped that we can target GABA to block granulocytic MDSCs and use the GABA analog pregabalin. So pregabalin, again, is a clinically used GABA analog that is important to manage neuropathic pain. When we treated our mice with this drug, tumor-bearing mice with pregabalin, and when we hoped that we would block the granulocytic MDSC activity, we actually observed an opposite outcome. So female mice that received this drug succumbed to disease earlier than males, earlier than vehicles. In contrast, we didn't see the same effect in male mice. As always, we repeated the experiments using NSG immunocompetent mouse models. And again, we didn't observe differences in survival outcomes, suggesting that pregabalin treatment was driving disease in females in an immune-dependent manner. So we looked at whether there's a role for granulocytic MDSCs. And we did so by getting these cells out from bone marrows and stimulating them with GABA. Stimulation with GABA led to moderate changes in the expression profile, and we ended up observing an increase in 130 genes. But this was unique to female granulocytic MDSCs. When we repeated the same experiment with male granulocytic MDSCs or monostic MDSCs from either sex, we observed that there were no differences in the expression profile. So therefore, we focused on this as GABA response in female granulocytic MDSCs and looked what genes were upregulated and which pathway that was linked to. So one important pathway that came up from this screen was antiviral innate immune response. To further validate what was unique to females, we compared the expression profile of GABA-treated granulocytic MDSCs from females and males, and we observed that complement 1Q was highly expressed in female granulocytic MDSCs that encountered GABA. So you guys might remember this from your undergrad course, which is one of the least favorite topics in immunology. The complement system is an important innate immune system. Just briefly to recap the complement system, there are 3 ways that we lead to complement activation, and the one we are referring to here is the classical pathway, which was initiated by complement 1Q. So complement 1Q forms complexes with C1R and C1S, and collectively this complex recognizes the antibodies that are circulating in patients in host, or apoptotic cells, which could be more relevant in the tumor microenvironment. This C1Q immune complex formation leads to the cascade of cleavage events and production of additional complement molecules such as C3A and C5A. So this is important for opsonization and fighting against infections, but there could be a role in the tumor microenvironment as well. Several studies suggested that endothelial cells and tumor cells express receptors for these complement molecules, and binding of these C1q, C3, or C5a to these receptors can lead to increased angiogenesis or can alter tumor cell phenotypes by increasing their proliferative capacity or stemness. So we wanted to understand whether complement 1q was indeed a relevant target. And we return back to our preclinical models. So we wanted to do this by an unbiased way. Therefore, we sorted out monocytic MDSCs, granulocytic MDSCs, and macrophages from tumor microenvironment based on markers that they express. We also sorted out monocytic and granulocytic MDSCs from peripheral blood of mice with tumors. So this analysis, which is single-cell analysis from these populations, led to the identification of 9 separate clusters. And when we looked at the gene expression levels, we observed that tumor-infiltrating myeloid-derived suppressor cells has significantly different expression profile than those in the peripheral circulation. We also again identified sex as a driver of expression profile, and different clusters were represented at different levels between males and females. So what genes were differentially regulated within these pathways, within these clusters? And we observed again that subunits of complement 1q were highly expressed by tumor-infiltrating granulocytic MDSCs, which are likely get exposed to GABA compared to any other cluster. I would like to highlight that IL-1 beta again emerges as a therapeutic target, which is expressed mainly by peripheral granulocytic MDSCs, and that really matched our earlier data, which was very reassuring. So since we again identified complement 1q as a therapeutic target, we wanted to understand whether these RNA levels changes correlate with a disease outcome, or whether it has physiological protein level outcome. And we did an ELISA to measure C1q levels between male mice and tumors and female mice with tumors. And collectively, we observed that female mice with tumors had significantly higher levels of complement 1q compared to male mice with tumors. There are transgenic mice that we can use, so that's what we did. And we collaborated with a researcher who had those. And we implanted C1q knockout mice or wild-type mice with tumors, with brain tumors. And in this setting, we observed that there was no difference in the survival duration of wild-type male mice with GBM tumors or C1q knockout mice with GBM tumors. In contrast, in females, we observed that C1q knockout female mice survive for a longer duration compared to wild-type littermates. So collectively, these results indicated that C1q knockout mice, C1q might be a tumor-promoting factor, specifically in females and not in males. We wanted to validate these observations in patients, and we did so by, again, looking into 2 publicly available databases, the Cancer Genome Atlas and Chinese Glioma Atlas. In this setting, we observed that C1q expression level is not predictive of outcome for males, but in females, we observed that high levels of C1q correlate with a worse outcome compared to patients with low levels of C1q expression. So why are we interested in C1q? Well, there are several reasons, but one of which is that, again, there's a clinically used inhibitor that we can repurpose. C1 inhibitor is an inhibitor of C1 esterase, and it is used for treatment of angioedema. So in very preliminary studies, we grabbed this inhibitor and tested in our in vitro in our bone marrow cultures. So we sorted out bone marrows and put them in MDSC polarizing conditions and included C1 inhibitor in our cultures as well. So in this setting, we didn't observe any phenotypic changes of a relative representation of monocytic MDSCs, granulocytic MDSCs, but what we observed was a functional change. At the very beginning of my talk, I mentioned that the main function of MDSCs is their ability to suppress T cells. So when we do a T cell proliferation assay, in which we co-culture MDSCs and T cells, and measure T cell proliferation by activating T cells and looking for their division rate, we observed that we could reverse the suppressive capacity of granulocytic MDSCs. So here the black bars are controls where we observe proliferation of T cells. Blue ones are the ones that are cultured with monocytic MDSCs, in which we observe a reduction in the proliferation capacity of T cells, and C1 inhibitor had no effect on this compared to vehicle control. But when we look into orange bars here, we observe that vehicle-treated granulocytic MDSCs are more capable of suppressing the proliferation of T cells compared to those that encounter C1 inhibitor. So this really brings up this hypothesis that we are currently pursuing. We think that the GABA produced by astrocytes or neurons in the tumor microenvironment functionally alter tumor-infiltrating granulocytic MDSCs, which express GABA-B receptors. We observe that this GABA-B receptor activity leads to increased C1q production, and this somehow drives brain tumors. And we also believe that this is a dominant mechanism in females, which is characterized by higher granulocytic MDSC activity. So what are we trying to do now? We are trying to understand the role of GABA in myeloid cell regulation, as well as in other immune cell types. We are trying to understand whether it's the GABA or GABAergic neuronal activity that leads to these functional changes. We are trying to, again, identify the cell intrinsic changes in granulocytic MDSCs upon exposure to GABA and pathways that are activated. So what about C1q? Yeah, we are very interested in understanding what it does in the tumor microenvironment. And we would like to obviously therapeutically the target and incorporate that as part of the standard of care in brain tumors. So with that, I want to leave ample time for questions. So I will acknowledge my postdoc mentor, Justin Latia from Cleveland Clinic, who was very supportive of my career and my transition as an independent investigator. We had a fairly large lab, and not all these people were present at the same time, but I had the privilege to work with these brilliant scientists across 5 years, I guess, that I was in Justin's lab. It takes a village to do high-impact studies, so it's really important to collaborate with clinicians and scientists with different expertise, and this is what happened. You know, you can see that there were many internal and external collaborators that help with different aspects of this project, and Antonio Arborone is my actually faculty mentor who is helping me to start my own lab. So we are a very new lab. I mentioned as earlier, we started our lab 3 months ago, and I was very lucky to be able to start working with two great scientists, Oriana Teran-Pumar, who started a month ago, and Asmita Patak will soon join. With that, before moving on to questions, I actually want to switch to Turkish and want to make some remarks. So those who don't speak Turkish, I apologize. Arkadaşlar, hepinize katıldığınız için çok teşekkür ederim. Benim söylemek istediğim, yaşadığınız zorlukları farkındayız. Biz özellikle yeni veya Türkiye'den ayrılalı çok olmayan bilim insanları olarak durumu farkındayız ve size elimizden geldiğince yardım etmek istiyoruz. Bunun hani ekonomik zorluklar olduğunu farkındayız, eğitimsel zorluklar olduğunu farkındayız, pandemi, daha sonra deprem nedeniyle. Bu yüzden eğer olur mu olmaz mı bilmiyorum ama Hakan Ceylan ve Murat Yıldırım'la bir platform başlatmayı deniyoruz. Dediğim gibi belki başarırız, belki başaramayız. Çok yeni bir oluşum bu. Ama bu platformla size staj olanakları yaratmak istiyoruz veya en azından sizi Türkiye'de bulunan veya yurt dışında staj olanaklarıyla eşleştirmek istiyoruz. Böyle bir misyonumuz var. Bu yüzden aktif olarak diğer araştırmacılara hem yurt içinde hem yurt dışındaki araştırmacılara ulaşıyoruz ve bir platform kurmaya çalışıyoruz. Yine bunun bir parçası olarak sizin başvurularınıza yardımcı olmak istiyoruz. Yani bunu daha eğer büyüyebilirsek tabii daha iyi bir şekilde yapabiliriz ama en azından hani o kurulana kadar da bize de ulaşabilirsiniz. Mesela CV hazırlama gibi bir yardımda bulunabiliriz veya özgeçmişinizi özgeçmişinizi hazırlamanıza yardımcı olabiliriz veya nasıl e-mail atılır, Amerika için ayrı, Avrupa için ayrı beklentiler oluyor. Bu gibi şeylerde size danışma için yardımcı olmaya çabalayabiliriz. Ekonomik zorluk tabii ki çok önemli bir etken burada. Bizim zamanımızda belki bu kadar ciddi bir ekonomik kriz yoktu. Sizin kadar etkilenmedik. Ama size maddi destek imkanları yaratmak istiyoruz. Ne yazık ki çoğu enstitüde staj için gelecek öğrencilere maddi bir yardımda bulunma olanağı yok. O yüzden bazı yerlere ulaşarak belki programlar oluşturmak ve staj için destek sağlamak gibi bir misyonumuz var. Dediğim gibi bu çok daha kurulma aşamasında olan bir şey. O yüzden ne kadar başarırız bilmiyorum. Ama sadece yanınızdayız. Bunu bilmenizi istiyorum. Ve belki bu kuruluna kadar mesela ben kendi adıma söyleyeyim. Elimden geldiğince size yardımcı olmak isterim. O yüzden her zaman bana Twitter'dan ulaşabilirsiniz veya her zaman bana e-mail atabilirsiniz. Okumamı istediğiniz bir şey varsa veya yorum yapmamı istediğiniz bir şey varsa ben yardımcı olmaktan çok gurur duyuyorum. Hakan ve Murat'ın da muhtemelen aynı şekilde hissettiğini düşünüyorum. Hepimiz çok farklı alanlardan gelen insanlarız. Farklı dünya görüşü, farklı politik görüşü, farklı sosyal görüşü olan insanlarız ama aynı çatında altında buluştuk. Hepimizin bilime olan inancı var ve hepimizin size yardımcı olmak gibi bir misyonu var. Bu da aklınızda bulunsun. O yüzden again, switching into English, I would be happy to answer any questions you have regarding the slides I presented today or anything else I can help with. Looking forward to the discussion. Thank you again for inviting me.
Merhaba hocam. Öncelikle desteğiniz için gerçekten çok teşekkür ederiz, minnettarız. Ben de şu anda İngilizce olarak devam ediyorum. I appreciate your wonderful presentation, Dr. Defne Bayık. If you wish, we can go to the question and answer section prepared by our team and consisting of questions from our participants.
Sure, we'd love to.
Okay. Our first question is, gonadal sex steroids are found in sex chromosomes and affect on neuroanatomy and neurochemistry. For this information, can we say glioblastoma treatments can be related to connectivity among neurobiological changes but not hormonal changes?
Yeah, so that's a wonderful question. We actually don't know the answer. Let me start with that. So they're not just in glioblastoma, but the sex differences are getting extensively studied across tumor types. And we actually know that androgen receptor signaling could play a potential role in the regulation of mostly T cells, to be honest, not myeloid cells, but T cells and their anti-tumorigenic function. That's something I am very much interested in studying because it's been shown that estrogen and progesterone, more so progesterone, is linked to neuronal regulation. So there could definitely be a link between hormones and their, direct effect on the immune system or in the microenvironment, and that could lead to potential outcome differences in glioblastoma. With that being said, I will also tell that the sex differences in incidence is considered across age range, meaning that even early puberty to very late in life, there is a difference between males and females, suggesting that the hormones may not driver. So I don't think, I know it doesn't answer the question all that well. I think bottom line is that we don't have a good idea how the whole equation works, and that's something I'm very much interested in studying.
Thank you for your answer. Next question is, the reason that you have been used GL261 type mice because they have highly immunogenic relativity to human GBM, right?
Actually, no. So I will tell you, if anything, GL21 model is not the best model. The reason being that it's a hypermutated mutated models. So GL261s are generated by random mutagenesis. And in patients, what we observe is that there are very limited mutations in patients with glioblastoma. The mutation burden is very low. So that's why it's not the best model to use. But that's also why we use multiple models in the lab, such as SB28 and KR158, which have targetable mutations that are identified in patients with glioblastoma. I will tell you, as in many cancers, modeling glioblastoma is very hard in mice. And that's kind of why we always try to validate whatever results we get in preclinical models. We try to validate in patients to confirm that those mechanisms are conserved between the species, and models are indeed good to study those mechanisms.
Thank you so much. I'm moving on with the next question. Hormonal estrogen support to men. High probability we see hormonal changes on sex-based behaviors, but in social cultural frame. These behaviors can be reduced for the very good treatment's efficiency.
Yeah, I guess if I understand the question right, the question is the impact of social sex behavior. I guess the difference between gender and sex, which I know we don't have the same concept in Turkish, toplumsal cinsiyet ve biyolojik cinsiyet. So again, I don't know how much roles does the hormones play in this equation. I don't think they are the major drivers. I think the chromosomal or epigenetic drivers are the main effectors of the outcome in the context of glioblastoma. With that being said, that's actually a very important point, because there are several studies that suggest gender differences are important for tumorigenesis as well. So one example I can give is that males are more likely to smoke than females, and that is a major contributor of lung cancer incidence. So how do you discriminate between gender behavioral differences versus biological sex? That's tough. I mean, I think in mouse models it's obviously easier because there are not many gender differences. Certainly there's differences in aggression, that kind of stuff, but not life choices that would affect the glioblastoma outcome. So role of hormones still under investigation. I can tell you that, again, we have a study under review. A friend of mine led that study and focuses on androgen receptor, androgen signaling, and glioblastoma outcome. But I don't know whether estrogen has a role and how that plays into behavioral aspect.
Thank you for your response. I'm moving on with the next question. Glioblastoma has male gender incidence bias, but is there currently clinical trial data examining differential effects of treatment between genders?
Yeah, so yes and no. That's what we are hoping to achieve. We are trying to initiate clinical trials by using this information. It's very hard to exclude the sex from a clinical trial because of regulatory reasons, but we are pushing. There's a very big push to account that when you measure treatment response. And there is no currently any, that's something we started doing, but there are no clinical trials that specifically measure sex differences in the context of glioblastoma. But recent studies for checkpoint inhibitors, again, which are getting widely used in lung cancer, melanoma, GI cancers, and those studies suggest that there is a difference in response rate based on sex, as well as there's a difference in the adverse reaction that is observed in patients. So clearly this is an emerging field, and we are just starting understanding how patients respond differently based on sex and what to expect.
Thank you for your answer. And next question is, how feasible is it today to independently investigate sex hormones and chromosomes for sex-dependent immunotherapy responses in GBM?
Yeah, that's a wonderful question. There are models to do that. And one such model is called 4-core genotype model. So 4-core genotype model is a model in which which there are four genotypes. One of them is XX, the other one is XY. The other two is that XY without the SRI gene, which is the gene that promotes testicular development. So those XY minus SRI mice don't have testes, but other than that, they are chromosomally X and Y. And the fourth genotype is XX plus SRI. So basically, they move the SRI gene to an autosome in those mice. So those mice are chromosomally female, but they do develop testes. So those models are very useful to dissect the role of sex chromosomes versus sex hormones. But I agree that they are very hard to maintain. They are getting used more frequently. But that's the one mechanism we can use. The other mechanism that we can use, which is more feasible, is basically doing ovarectomy and castration and see how that could affect the outcome. But when we do that, that's obviously not the perfect model, in the sense that you do reduce the circulating hormonal levels, but you do not alter epigenetic or imprinting changes that happened early on life that is the result of exposing to sex chromosomes. But that's still a way, at least, to study the relative contribution of sex hormones versus chromosomes.
Thank you for your answer. Another question is, can sexually dimorphic genes play a role in stratifying survival outcomes in a large GBM patient dataset?
Certainly. I mean, I don't think anybody has done that, but that's certainly a possibility. And that's kind of from answering that from an immune perspective. We know that, for example, X chromosomes express many immunological genes. One such example is FOXP3, which is important for development of regulatory T cells, and toll-like receptor 7, which is important for innate immune regulation. And in fact, toll-like receptor 7 is one of the genes that escape dosage inactivation, meaning that females have higher levels of toll-like receptor 7 compared to males. So it's certainly a thing, something we should consider. We should look more deep into whether sex differences, sexually dimorphic genes, is a relevant contributor of patient outcomes. The other thing I will briefly touch on is that both X and Y chromosomes do express epigenetic modifiers. So even if we don't see maybe chromosomal level changes, these epigenetic modifiers are likely to regulate gene expression levels differently and lead to different outcomes. But we should definitely look into it.
Thank you so much. I continue with next question. The tumor microenvironment occurs by specific interaction between the tumoral and the host biological factors. For these relationship conditions that happen during medical treatments, is there a way to decrease tumoral function and parallel that increase the biological factors mechanism efficiency?
Is there a way to decrease tumoral function and in parallel increase the biological factors mechanism efficiency? Yeah, that's an intriguing question. I will say yes. And I don't think we know that. That's the bottom line of it. But again, going back to the GABA story that I shared with you, so that we initially tried out GABA analogs, hoping that would be the mechanism, but which didn't turn out to be the case. But there are GABA inhibitors that we can use, and that would likely to decrease the tumoral function, but also affect the biological mechanism. But we haven't tested that yet. So if we use, for example, GABA production inhibitors or receptor antagonists, maybe we cannot do both.
Yeah, thank you so much. Do sexually dimorphic genes tend to enrich for immunological signatures in both gender?
Yeah, I think that kind of, we touched on that a bit, that there are definitely immunological genes that are related to sex. But we understand that better in the context of autoimmune diseases. Again, going back to toll-like receptor 7, it's been linked to prevalence of autoimmune disease in females. We need to come up with those signatures for patients with tumors as well.
Okay. I appreciate for your answer. Another question is from YouTube. Does identifying predictive biomarkers of immune checkpoint inhibitors response for sex bias can be any benefit to cancer therapy?
100%, yes. The only problem in the context of glioblastoma is, as I briefly mentioned, that the checkpoint or the immune therapy strategies have failed in glioblastoma. So when you have a response rate of 2%, that's kind of not a good outcome to do it. But definitely for lung cancer, melanoma, that's something that should definitely be further pursued.
Thank you very much for your response. Another question is, considering the data of the Cancer Genome Atlas revealing different sex patterns and association to multiple cancer types, what is the hardness of coordinating a big amount of data?
Yeah, the biggest hurdle is how you collect the data, right? This data come across institutes, and you also don't know what this history of these patients, how the data is collected, how the specimens are processed. So I think the lack of multi-institutional standardized things is the main problem handling large data sets. We try to overcome that by generating our data. We have a large number of patients that are going through treatment in our institute, and we are. I mentioned that my faculty mentor he's very much interested in doing single cell sequencing with those patients and identify gene signatures that could correlate with outcome or identify treatment opportunities based on that signature. So that's what we'll keep doing. I think we'll generate our own large data to do so.
Okay, thank you so much for your answer. I'm moving over to the next question from YouTube. Nowadays, do we know how each of the MDSC subtypes contributes to pro-disease processes in men versus women will provide?
Yeah, we do and we don't. So I guess the question is how that is relevant in patients. So we know that they serve as biomarkers in many different tumor types. We know that each tumor type has slightly different association. For example, in pancreatic cancer, granulocytic MDSCs seem to be more important than monocytic MDSCs. In livers, the other way around, monocytic MDSCs seem to play a more bigger role than granulocytic MDSCs. So I think by studying the patient data more on different tumors, we can get a better understanding of how each subtypes interact with these different disease processes, environment, and tumors, and how that could be an important driver of the disease.
Thank you very much. Another question is, there is a complex interaction among sex-based behaviors, hormones, and genes, and as a result of this could have affected the adaptive immune system. Is it possible to collect and coordinate all of the efficient factors in one spot?
Patients are very complex, right? So it's very hard to I don't think we have all the data to make those correlations. So when patients walk into clinic, we don't, for example, measure their androgen levels or estrogen levels, and we don't see how that could play a role. But that's what we are hoping to do, in a sense that if we could identify female-male specific disease biomarkers, for me that's immune, probably for others that's like different contributors, different mechanisms, we could potentially address that. But I think it's just very hard to bring all this data together due to the patient variation, which is something we oversimplify using mouse models.
Yeah. Thank you so much. Another question is, apart from gender-related treatment response, is it necessary to evaluate pediatric patients separately in GBM?
100%. Sorry.
Are there differences in in tumorigenesis in adult and pediatric patients?
Yes, 100%. And all the data I presented today is for adult brain tumors, not pediatric brain tumors. Pediatric brain tumors also have tendency to show sex differences. Again, it's unfortunately more common in male kids than female kids, but there are different driver mechanisms. It's a very much understudied area. We don't really actually have a good understanding of immune infiltrates in pediatric brain tumors, but there are definitely different mechanisms in place. So with those young adult and pediatric tumors are a completely different field of study.
I appreciate for your answer. Next question is, in the light of all this information, could sex consider as a biological variable for development of effective immunotherapeutic strategies against GBM as well as other cancers?
Yes, and that's what we try to do with the IL-1 beta as well. So the drugs we usually use are, again, clinically approved drugs for different purposes, so that we can basically repurpose them for anti-tumor. And we did try to initiate one such trial using IL-1 beta inhibitors. But the challenge is that you need an industry partner who is willing to do that. I'm happy to discuss the business aspect of the stuff, but what I can tell you is that we were about to start a clinical trial doing just that, but then the company backed down because their priorities have changed. So I think overall, is it possible? Yes, but there are some challenges to make that happen.
Okay, thank you. And next question is from YouTube. Functional and targetable sex differences present within certain myeloid populations. Can GBM infiltrating T cells actually mediate sex difference in overall survival?
Yes, the answer is yes. And I don't know whether you guys are familiar with the bioarchive. This is where people deposit their preprints before pre-review, which is a great way of sharing science freely before pre-review. So that's kind of the catch there. So not all the papers are great, but certainly that's the platform we commonly use to get the data out there. And Julian Lee is the first author of that paper that focuses on T cells and sex differences in the context of glioblastoma. I had the privilege to work with her as a postdoc. She was a postdoc in the same lab as I was, and you could find more information there.
Thank you. And another question is, we can examine androgen receptors due to the location of AR gene on the X chromosome. Depending on the different location, types of AR gene can cause a higher prevalence GBM in men. Based on the facts, can be found antagonist-based treatments for men and women.
So is it the androgen receptor and how it affects how it can be combined with treatment? Yeah, there are androgen receptor blockers, right? So I know that there are studies now in BRAF melanoma that incorporate androgen receptor antagonists to increase the efficiency of immune therapies. Jennifer Vargo is the lead senior author on that paper. In glioblastoma, several studies have suggested that tumor cells can express androgen receptors, and that could be an important factor. We have not studied androgen receptor signaling so far, but that's a good point that we should definitely look into.
Thank you so much. We have last question. What advice would you give to students studying in scientific fields?
Yeah, so is the person who asked the question Turkish?
You can answer both. Yeah, it doesn't matter. Yeah, you can answer.
Okay, then I'll switch to Turkish. Yeah. Ne gibi şöyle scientific network'ünüzü genişletmeniz gerekiyor. Çünkü aslında akademinin içinde olunca insanın anladığı şeylerden biri sizin arkanızı kollayan birinin olması ne yazık ki çok önemli. Belki bunu hiç düşünmüyoruz ama danışmanınız olsun veya yurt dışında tanıdığınız biri olsun. Onların size verecekleri referans mektupları her şey için çok önemli. Bağlantı kurmak çok önemli. Bunun için hani siz zaten buraya gelip hani konferansa katılarak yapıyorsunuz. Virtual olsa bile bu yani mesela bana bir gün yazarsanız işte ben sizin işte buradaki konuşmanızı şey yapmıştım, bana mektup verir misiniz? Ben tabii falan şeklinde yaklaşırım ama en önemli şey ne yazık ki bağlantı kurmak. Aynı zamanda tabii yapacağınız kendinizi bilim açısından geliştirmeniz de çok önemli. Yeni teknikleri öğrenmek veya en azından hani teknik olarak bilmeseniz bir de hani alanlar nereye ilerliyor işte ne bileyim single cell sequencing mi, CRISPR mı, işte spatial transcriptomics mi? Hani hangi alanlara doğru böyle bir şey olduğunu anlamak ve o alanlara yoğunlaşmak gerçekten çok yardımcı oluyor. Çünkü o sayede daha hani daha iyi makale basabiliyorsunuz, daha etkili bilim yapıyorsunuz. Ama verebileceğim en büyük advice, sizi destekleyecek insanlar bulun. Mentorlarınızı çok dikkatli seçin. Sizi destekleyeceklerinden emin olun ne yapacaksınız. Yine kendimden örnek verecek olursak mesela ben doktoraya aslında Türkiye'de başladım. Ama benim danışmanım beni NIH'e yolladı doktoramı bitirmem için. Ve benim için o çok ilk kapıyı o şekilde açtı. Ondan sonra belki kalma niyetim yoktu. Hani hayat kalmaya şey yaptı, beni doğru itti. Ama onun o verdiği destek çok önemliydi. Mutlaka sizi destekleyecek birilerini bulun ve mümkünse sizi destekleyecek bir sürü insan bulun. Size verebileceğim en büyük şey bu olur.
I will switch to Turkish now. Hocam öncelikle tavsiyeleriniz için gerçekten çok teşekkür ederiz. Bizim için çok önemli. Ayrıca daha önce verdiğiniz destek için de teşekkürler. Yani biz Türk genç olarak gerçekten bunlara çok ihtiyacımız var. O yüzden gerçekten çok sağ olun. And I'm switching to English now. On behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us and for this great informative presentation.
Thank you very much for inviting me again, and looking forward to seeing you guys down the road. You are doing some great stuff here.
Thank you. I wish you healthy days.
You too. Bye bye.